Hi, good evening. In this session, I am going to discuss electron transport system, which is the final stage of the aerobic respiration. Already we have discussed glycolysis and Krebs cycle in the previous lessons. And now I am here to discuss electron transport system, otherwise ETS. What is electron transport system? In electron transport system, what happens? Actually here, oxidative phosphorylation takes place. What is oxidative phosphorylation? Formation of ATPs in the presence of oxygen is said to be oxidative phosphorylation, or formation of ATPs by utilizing the energy released by reduced coenzymes that are produced in either glycolysis or connecting link reaction or Krebs cycle. So what are the reduced coenzymes like NADH2 and FADH2? Actually NAD, FAD are the coenzymes that are reduced, so that are called reduced coenzymes. Now NADH2 and FADH2, they have reducing equivalents. That NADH2, FADH2 that are produced during the Krebs cycle or connecting link reaction or glycolysis undergo oxidation in electron transport system. And this electron transport is coupled with the formation of ATPs in the presence of oxygen. Hence it is said to be oxidative phosphorylation that takes place on the inner mitochondrial membrane where the oxysomes are located. Otherwise F1, F0, F1 particles are elementary particles. Here in electron transport system, various intermediate electron carriers involved to transfer the electrons from NADH2 or FADH2 to terminal electron acceptor oxygen. So these electron carriers, some of them are located as complexes and some of them are as free electron carriers, otherwise mobile electron carriers. So there are four complexes of electron carriers: complex 1, complex 3, complex 2, complex 4. These four complexes are made up of electron carriers, whereas the complex 5 is also there, but it is not the complex of electron carriers, but it is the complex of ATPase or ATP synthetase enzyme, which is responsible for the synthesis of ATPs. Let us discuss the mechanism. So here electron carriers are located in four complexes, known complex 1, complex 2, complex 3, complex 4. Apart from that, these four complexes, some electron carriers are present in the inner mitochondrial membrane. There are two. You can see this. One is ubiquinone and other is cytochrome C. Ubiquinone is present in the inner mitochondrial membrane, whereas the cytochrome C is present on the outer surface of the inner mitochondrial membrane. Actually, this red band is inner mitochondrial membrane. Left side the inner mitochondrial membrane, the space will be there, perimitochondrial space, the space which is present in between the outer mitochondrial membrane and inner mitochondrial membrane. And right side this inner membrane, all this is a matrix. So this is matrix, it is inner membrane, and this is perimitochondrial space. Cytochrome C is present on the outer surface of the inner membrane, whereas the mobile electron carrier ubiquinone is present in the inner mitochondrial membrane. So these two are the mobile electron carriers, while all other are located in different complexes like this. Different electron carriers are present in the electron transport system. They are arranged in an orderly manner in inner mitochondrial membrane. If you see the complexes, Coming to the components of the complexes. If we take the complex 1, it consists of FMN and ferrous sulfur proteins. If we take the complex 3, it consists of cytochrome C1 and cytochrome B and ferrous sulfur proteins. If you see the complex 2, it consists of FAD and ferrous sulfur proteins. If you see the complex 4, it consists of 2 copper centers and cytochrome A and cytochrome A3. And complex 5 is made up of enzyme that ATP synthetase. Almost all the complexes they have enzymes. If you see the complex 1, it consists of NADH2 dehydrogenase enzyme. You see the complex 3, it consists of cytochrome reductase enzyme. You see complex 2, it consists of succinic dehydrogenase, otherwise FADH2 dehydrogenase enzyme. You see the complex 4, it consists of cytochrome oxidase, which oxidizes the cytochrome. Enzyme is present here, and here however ATP synthase is present. Like this, these are the various components of the different complexes that involve in the electron transport system in respiration. Coming to the mechanism of oxidation of NADH2 or FADH2. Actually, how it takes place oxidation. You suppose NADH2 which are produced in connecting link reaction, they are produced, even in glycolysis they are produced, even in Krebs cycle also they are produced. You take one NADH2. When NADH2 enters the electron transport system, NADH2 gets oxidized. How NADH2 is oxidized? By losing the electrons and protons, otherwise hydrogens. Actually, NADH2 is a reduced coenzyme. Its actual form is NAD+. There it was reduced. Now again, it undergoes oxidation. It will be oxidized to its original form NAD+. And electrons and protons are released. That electrons are transferred to FMN. This reaction takes place under influence of NADH2 dehydrogenase, which removes the hydrogens or reducing equivalents from the NADH2 that are transferred to the FMN. FMN in turn transfers the electrons to the ferrous sulfur protein. Finally, the ferrous sulfur protein transfers the electrons to ubiquinone, which is a mobile electron carrier which is present in between the complex 1 and complex 3. During the movement of electrons, kinetic energy will be released, and this is useful to pump the protons from mitochondrial matrix to perimitochondrial space. During the transport of a pair of electrons from NADH2 to FMN or complex 1, 4 protons are pumped out from mitochondrial matrix to perimitochondrial space. Regarding this calculation, various opinions are there. This is a $1 million question. Okay, let us discuss at the ending. Some workers said that only 2 protons are pumped out, and according to some workers 4 protons are pumped out. Okay, we take one model. Here 4 protons are pumped out. So protons are pumped out into perimitochondrial space, whereas the electrons are transferred to the ubiquinone. Ubiquinone is a mobile electron carrier which accepts one electron at a time. If it accepts one electron, that becomes UQ minus, that is semi-quinone. Then it accepts 2 protons also from the mitochondrial matrix and becomes UQH2. So when it accepts one electron, it partially reduced. When it accepts 2 protons also from the mitochondrial matrix, it will be completely reduced. Now this completely reduced form UQH2 is called ubiquinol, and partially reduced one is called semiquinone, and the version is called ubiquinone. Ubiquinone semi-quinone, ubiquinol, fully reduced, completely reduced form of the ubiquinone. Now this ubiquinone, it transfers that whatever the reduced form, completely reduced form ubiquinol, transfers the electrons to the complex 3. So that is the function of the ubiquinone, which transfers electrons from complex 1 to complex 3. Now the electrons are transferred to first cytochrome B, components of the complex 3, cytochrome B, ferrous sulfur protein and cytochrome C1. Initially electrons are transferred to the cytochrome B, from there electrons are transferred to the ferrous sulfur protein, and finally electrons are transferred to the cytochrome C1. And cytochrome C1, it transfers its electrons to the cytochrome C, means cytochrome C will be reduced. Not only cytochrome C, every electron carrier in the electron chain will undergo reduction and subsequent oxidation. If it accepts electrons, it undergo reduction. If it releases electrons or transfers electrons to the adjacent electron carrier, it undergoes oxidation. So here cytochrome C is reduced under influence of cytochrome C reductase enzyme, which is present in the complex 3. Now electrons are transferred to the cytochrome C. From there electrons are transferred to the complex 4. Here that is the function of the cytochrome C. Cytochrome C is a mobile electron carrier which is located on the outer surface of the inner mitochondrial membrane. In between complex 3 and complex 4, it transfers electrons from complex 3 to complex 4. In complex 4, 2 copper centers are present, and on cytochrome A, cytochrome A3 also present. Initially, the electrons are transferred from cytochrome C to copper center. From there, electrons are transferred to cytochrome A. From there, electrons are transferred to cytochrome A3. Again, the electrons are transferred to cytochrome, copper center finally. Now, from this copper center, electrons are transferred to oxygen. Actually, here the terminal electron acceptor in the aerobic respiration is oxygen. So, half molecule of oxygen is present, and that takes the electrons and gets reduced, and it also accepts it to protons. It draws 2 protons from the mitochondrial matrix, and it forms H2O. So like this, every time either one NADH2 or FADH2, if it is oxidized, half molecule of oxygen is consumed. Totally, how many reduced coenzymes are there? The 12 reduced coenzymes are there. Where in the glycolysis 2, in the connecting link reaction 2, 4 NADH2s, and in mitochondrial matrix 6, 10, and 2 FADH2 also present in mitochondrial matrix. Together, 12 reduced coenzymes are there. Of them, 10 are NADH2s and 2 are FADH2s. Means, you suppose 12 times electron transport system is being operated. Every time electron transport system it is operated, half molecule of oxygen is consumed. So we have 12 reduced coenzymes, 12 into halves, 6 molecules of oxygen is consumed. That is why in aerobic respiration we write 6O2, glucose plus 6O2, right. So actually oxygen is neither consumed in glycolysis nor in Krebs cycle or anywhere else. The role of oxygen is restricted to electron transport system. It acts as electron acceptor, terminal electron acceptor. That is why we call this aerobic respiration. The aerobic respiration means here the reducing equivalents are transferred from organic compound to an external source, that is nothing but oxygen. When electrons are transferred to oxygen, oxygen gets protons from the mitochondrial matrix and it is converted to water molecule. So every time when NADH2 or FADH2 is oxidized in electron transport system, half molecule of oxygen is consumed, so 12 into half 6, and 1 molecule of water is formed, clearly seen here. So 12 into 1 water molecule, 12 water molecules are resulted. So that is why in the aerobic respiration reaction we represent we write 12 water molecules right side the arrow mark, because water molecules are resulted in electron transport system, and these water molecules together constitute metabolic water. So how much metabolic water is produced in aerobic respiration? 12 molecules of water is produced. Every time when NADH2 or FeIDH2 is oxidized, one water molecule is produced, half molecule of oxygen is consumed. That is why 12 into halves, 6 molecules of oxygen is consumed in aerobic respiration, and 12 into 1, 12 water molecules are produced in aerobic respiration. Okay. During the transport of electrons through various intermediate electron carriers from NADH2 to finally oxygen, how many protons are pumped into the perimitochondrial space is the question. So that is needed here. During the transport of a pair of electrons through various electron carriers finally to the oxygen, at 3 points protons are pumped out. So at the complex 1, 4 protons are pumped out. At the complex 3, 4 protons are pumped out. At the complex 4, 4 protons are pumped out. Sorry, 2 protons are pumped out. Together 4 plus 4 plus 2, 10 protons are pumped out from the mitochondrial matrix into the perimitochondrial space. So this is mitochondrial matrix, and this is perimitochondrial space. Now, this pumping of protons from mitochondrial matrix into the perimitochondrial space leads to the establishment of a concentration gradient in between the two layers. So here proton concentration is decreased, here proton concentration is increased because of the pumping while the transport of electrons. So this leads to the establishment of a concentration gradient. So a proton gradient is established in between two surfaces or two places, and a membrane potential is developed. This proton gradient and membrane potential together create a proton motive force, PMF. Proton motive force is created. Now what happens? As usual, here the concentration of protons is increased, here decreased. Now from high concentration to low concentration, protons flow back. So we know very well, movement of ions from higher concentration to lower concentration. This is facilitated by this F0, F1 particles. So here proton concentration high, here low. So from high to low, now the protons will flow back, breaking the concentration gradient. So here F0, this is the protein complex which is present inside, whereas the F1, it comes out into the mitochondrial matrix. F0 protein complex, it acts as proton channel which facilitates the movement of protons from perimitochondrial space into the mitochondrial matrix. When the protons are flowing back through the proton channel, they generate kinetic energy. That energy will rotate this rotatory instrument F1. It is also a protein complex which protrude outside the inside the inner membrane, means into the matrix. Now protons are flowing back through the proton channel. When protons are flowing back from perimitochondrial space into the mitochondrial matrix, energy is released, and that energy is used for the synthesis of ATPs. So ADP and inorganic phosphates, they combine, resulting in the formation of ATP. ATP is synthesized in the presence of oxygen. That is why it is called oxidative phosphorylation. So how many protons should flow back from perimitochondrial space into the mitochondrial matrix to form one ATP is the question. So this question is very worthful, $1 million worth. Some workers say 2 protons are enough and some workers say 3 protons are needed and some other intellectuals say 4 protons are needed. So different opinions are there. Okay, we discuss one opinion that 3 protons are needed for the formation of 1 ATP. If it is true, how many protons are pumped out during the transport of electrons from one NADH2 or during the oxidation of one NADH2? How many protons are pumped out from the mitochondrial matrix into the perimitochondrial space? 4, 4, 2, 10. So 10 protons are pumped out. If 10 protons are pumped out, what I said, 3 protons are needed. 3 protons should flow back from perimitochondrial space into the mitochondrial matrix to form 1 ATP. For 1 ATP, if 3 protons are needed, 10 by 3, that equal to 3 ATPs. That is why it is said that 1 NADH2 is equal to 3 ATPs. You might have read anywhere. Why 1 NADH2 equal to 3 ATPs? Because during the oxidation of 1 NADH2, 10 protons are pumped out into the perimitochondrial space from the mitochondrial matrix, leading to the establishment of a concentration gradient. Again, the protons will flow back from higher concentration to lower concentration. During movement of protons or flow back of protons, kinetic energy will be released. By using that kinetic energy, ATPs are synthesized. Minimum 3 protons should flow back from perimitochondrial space into the mitochondrial matrix to form 1 ATP. So, during the oxidation of an NADH2, 10 are pumped out. All the 10 flow back, 3 ATPs are formed. That is why 1 NADH2 equal to 3 ATPs. Okay, coming to the oxidation of FADH2. FADH2 does not enter the electron transport system through the gateway. Through complex 1, it does not enter. It enters the electron transport system through the complex 2. So, where it is complex 2? So, here it is complex 2. Yeah. When it enters through the complex 2, the electrons are transferred to ubiquinone, again as usual from ubiquinone to complex 3, again from complex 3 to complex 4. So, electrons are not transferred to complex 1. So, here these protons, 4 protons we miss in the case of FADH2. These 4 protons are not pumped out from the mitochondrial matrix into perimitochondrial space, because if this enters into the electron transport system through complex 1, then that 4 will be pumped out. But here FADH2 entering into the electron transport system through this complex 2, not complex 1. So, these 4 we miss and remaining is as usual. So, during the oxidation of one FADH2, only these 4 protons pumped out and these 2 protons pumped out. Only 6 protons are pumped out into the perimitochondrial space from the mitochondrial matrix. If these 6, if they come back, 6 protons by 3 protons, as I said, minimum 3 protons are needed for the formation of 1 ATP. In the case of FADH2, during the oxidation of FADH2, only 6 protons are pumped out from mitochondrial matrix into the perimitochondrial space. If all those 6 flow back, only 6 protons by 3 protons means we get only 2 ATPs. If it is NADH2, 10 pumped out, 10 flow back, 3 ATPs are generated. But in the case of FADH2, only 6 are pumped out protons. If all the 6 flow back, 6 by 3, 2 ATPs. That is why one FADH2 is equal to 2 ATPs, whereas one NADH2 equal to 3 ATPs. Because NADH2 enters into the ETS through the complex 1, whereas the FADH2 enters ETS through the complex 2. So, that is the difference. Because of that, the number of protons that are pumped out into the perimitochondrial space from the mitochondrial matrix differs. So, that will results in different varying number of ATPs. Okay. Now we came to a conclusion that one NADH2 equal to 3 ATPs and one FADH2 equal to 2 ATPs. This is okay. But coming to the cytosolic NADH2, which is produced in the cytoplasm, which reaction takes place in cytoplasm? Which step of the aerobic respiration takes place in cytoplasm? Glycolysis. In glycolysis, how many NADH2 are formed? 2 NADH2 are formed. So in that case, some change will be there. Sometimes cytoplasmic NADH2 or cytosolic NADH2 equal to 2 ATPs, sometimes 3 ATPs. So that depends on the shuttles in eukaryotes like malate aspartate shuttle and glycerol phosphate shuttle. That is not possible now to discuss here. I will make one another video regarding that topic. Okay, you suppose NADH2 equal to 3 ATPs. Even the cytosolic NADH2 also equal to 3 ATPs. In general, NADH2 equal to 3 ATPs. But cytosolic NADH2, there sometimes it is equal to 2 ATPs, sometimes it is equal to 3 ATPs. So depending on the schedules, we discuss it later. Now you suppose even the cytosolic NADH2 is equal to 3 ATPs. Then let us calculate with the total how many ATPs are formed in aerobic respiration. You see this, you take this. Now you know that one NADH2 equals to 3 ATPs and one FADH2 equals to 2 ATPs. So by considering these concepts, let us try to calculate the total number of ATPs that are generated and the net gain of ATPs in aerobic respiration. We know if you take glycolysis, there 2 NADH2 are formed and 4 ATPs also formed directly, that is substrate level phosphorylation. Substrate level phosphorylation means formation of ATPs at the level of substrate itself. Where the metabolism occurring, there itself ATPs are formed. And whatever we have discussed until now is oxidative level phosphorylation, OLP. In the presence of oxygen, the reduced coenzymes are oxidized, resulting in the formation of ATPs. This is OLP, that is SLP. SLP means substrate level phosphorylation. Through this substrate level phosphorylation, 4 ATPs are generated there in the glycolysis. And 2 NADH2 are there. We know that NADH2 equal to 3 ATPs. 2 NADH2 into 3 ATPs equal to 6 ATPs. 6 ATPs are produced through oxidative level phosphorylation and 4 ATPs are produced through substrate level phosphorylation. Together 10 ATPs. 10 ATPs. From the 10, 2 are consumed in the glycolysis. We have invested 2 ATPs. One is for the phosphorylation of the glucose into glucose 6-phosphate and other is for the phosphorylation of fructose 6-phosphate into the fructose 1,6 bisphosphate. So here total 10 minus 2 consumed. So 8 is the net gain. When if cytosolic NADH2 equal to 3 ATPs. So that is in malate aspartate shuttle. I discuss in one another video. Now in malate aspartate shuttle, here net gain is 8. Connecting link reaction. There how many NADH2 are there? 2 NADH2. 2 NADH2 into 1 NADH2 equal to 3 ATPs. 2 NADH2 into 3 ATPs equal to 6 ATPs. Yeah. Coming to the Krebs cycle, how many NADH2 are there? 6 NADH2 are there. 6 into 3 ATPs, that equal to 6 into 3, 18 ATPs are formed. And 2 FADH2, each FADH2 equals to 2 ATPs. 2 into 2 ATPs equal to 4 ATPs. And however, in the Krebs cycle also, through substrate level phosphorylation, 1 ATP is generated per 1 cycle. 2 Krebs cycles are operated, 2 ATPs are produced. Actually, GTPs are produced subsequently that are converted to ATPs. So, here also again SLP. Through substrate level phosphorylation, 2 ATPs are resulted. So, now you calculate how many are there? There 8, you leave it 6 plus 18. 6 plus 18, 24. 24 plus 4, 28 plus 2, 30. So, in total 30 ATPs in Krebs cycle and connecting link, and in the glycolysis itself net gain is 8. Together plus 8. Together 38. Actually, total number of ATPs that are produced in aerobic respiration 40. If it is malate aspartate shuttle. Out of 40, 2 are consumed in the glycolysis. So the net gain becomes 38. If it is glycerol phosphate shuttle, I am not going to discuss the details here, just I am mentioning the name. If it is glycerol phosphate shuttle, there one NADH2 equal to not 3 ATPs but 2 ATPs, hence here only 4 ATPs, 4 plus 4 8 ATPs, 8 minus 2 6 ATPs, there 6, and here remaining same, here 30 ATPs, 30 plus 6 ATPs equal to 36 ATPs. So total number either 40 or 38. Net gain 38 or 36. If it is malate aspartate shuttle, the total number of ATPs produced in aerobic respiration 40. Number of consumed ATPs 2, 40 minus 2, 38 is a net gain of ATPs in the case of malate aspartate shuttle. And in the case of the glycerol phosphate shuttle, the total number of ATPs that are produced in aerobic respiration 38. Consumption 2, same, 38 minus 2, 36. So 36 or 38 net gain, 40 or 38 total number of ATPs that are produced in aerobic respiration. Okay. The malate aspartate shuttle and glycerol phosphate shuttle I will discuss in next video. So here, however, 12 times electron transport system is operated. This is an assumption actually. Here 12 times operated, every time half molecule consumed, half molecule of oxygen, so 12 into half, 6 molecules of oxygen consumed in aerobic respiration, and every time one water molecule is produced, 12 into 1, 12 water molecules are produced. That is why we mention the general equation of the aerobic respiration.